This classification of neurons, part 1. Neurons are classified as per 3 criteria: polarity, relative length of axons and dendrites, and shape of the cell body. According to polarity, they are pseudo-unipolar, bipolar, or multipolar. According to relative length of axons and dendrites, they are Golgi type 1 or type 2 neurons. According to shape of the cell body, they are pyramidal cells, fusiform or pyriform. Other types of neurons are stellate cells, glomerular cells, amacrine cells. According to function, sensory neurons, motor neurons, and interneurons. Sensory neurons carry impulses towards CNS found in dorsal root ganglion. They are unipolar. Motor neuron carries impulses away from CNS found in ventral root ganglion. They are multipolar. Interneurons are exclusively within CNS. Classification of neurons, part 2. Neurons are classified as per 3 criteria: polarity, relative length of axons and dendrites, and shape of the cell body. According to polarity, pseudo-unipolar, only one process, example, dorsal root ganglia of spinal nerves. Bipolar neurons are found in retina and olfactory epithelium. Multipolar, most common type, found in peripheral motor nerves. According to relative length of axons and dendrites, Golgi type 1 neurons have long axons, example, Purkinje cells of cerebellar cortex. Golgi type 2 neurons have short axons and star-shaped. According to shape of cell body, pyramidal cells in motor cortex of cerebrum, fusiform cells in motor cortex of cerebrum, pyriform cells, Purkinje cells of cerebellum. Other types: stellate cells of cerebellar cortex, glomerular cells, granule cells of cerebellar cortex, amacrine cells in retina. Neuroglia, part 1. Let's study introduction, structure, classification, and functions of neuroglial cells. Neuroglia provide structural and functional support to neurons, do not propagate nerve impulses, 5 to 10 times more numerous than neurons, and constitute more than half of brain's weight. Structure: highly branched, occupy spaces between neurons, and provide suitable microenvironment for neuronal activity. Classification: according to structure, microglia and macroglia. According to origin, microglia are derived from mesoderm and macroglia neuroectoderm. According to location, CNS, astrocytes, oligodendrocytes, ependyma, and microglia. Peripheral nervous system, Schwann cells and satellite cells. Functions: provide structural and functional support to neurons, form blood-brain barrier, phagocytosis, produce cerebrospinal fluid, and myelination. Neuroglia, part 2. Let's study neuroglia in CNS, that is astrocytes, oligodendrocytes, ependymal cells, and microglia. Astrocytes are largest and most numerous. They are star-shaped cells with many dendrite-like process. Terminal ends form foot process. These surround capillaries and form perivascular sleeve. Sleeve and capillary endothelium form blood-brain barrier. Types: protoplasmic astrocytes in grey matter, fibrous in white matter. Oligodendrocytes are smaller than astrocytes. They have fewer cytoplasmic process, form myelin sheath around nerve fibers in CNS. Ependymal cells line the ventricles of brain and central canal of spinal cord. Microglia are smallest neuroglial cells. Their function is phagocytosis and they are derived from mesoderm. Neuroglia, part 3. Let's study neuroglia in peripheral nervous system and clinical correlation. Satellite or capsular cells. These surround cell bodies of neurons in sensory and autonomic ganglia. They provide structural support and nutrition to neuronal cell bodies. Schwann cells, also called neurolemmocytes, form myelin sheath around peripheral nerve fibers. They also form neurolemma around all nerve fibers of PNS. Clinical correlation: gliosis. It occurs in response to infection, trauma, or stroke. Microglia migrate to damaged area and perform phagocytosis. Damaged neurons are replaced by proliferating astrocytes. This process is called gliosis.
Glioblastoma multiforme is most fatal brain tumor, arises from astrocytes, very poor prognosis. Life expectancy is 2 to 3 months.
Topic is typical spinal nerve part 1. Let's study introduction, roots, formation and division, posterior and anterior ramus. Typical spinal nerve is formed by two roots, anterior and posterior. These emerge through intervertebral foramen, and spinal nerve is a mixed nerve. Roots. Anterior or ventral root is motor in function, arises from anterior on cells. Posterior is sensory in function, contains dorsal root ganglion. Anterior and posterior roots unite to form nerve trunk, which divides into posterior and anterior ramus. Posterior ramus curves backward around the vertebral column, supplies deep muscles of back and skin, divides into medial branch and lateral branch. Anterior ventral ramus runs anterolaterally around the body wall, supplies the muscles and skin. In thorax, it supplies intercostal muscles. Branches are lateral cutaneous nerve and anterior cutaneous nerve, which supply the skin of thoracic wall. Is typical spinal nerve part 2. Let's study gray ramus communicans and its sympathetic distribution, white ramus communicans, plexus formation. Gray ramus communicans. Soon after formation, ventral ramus receives gray ramus communicans from sympathetic trunk ganglion. It contains postganglionic sympathetic fibers. Sympathetic distribution, blood vessels, sweat glands, erector pili muscles. White ramus communicans is present in all thoracic nerves and upper two lumbar nerves. Connects ventral ramus to sympathetic ganglion. Has preganglionic sympathetic fibers which arise from lateral gray horn cells. Relay in sympathetic ganglion. Plexus formation. Except thoracic nerves T2 to T12, ventral rami form plexus. Major nerve plexus, cervical plexus C1 to C4, brachial plexus C6 to T1, lumbosacral plexus L1 to S3. Differences between sympathetic and parasympathetic nervous system. Sympathetic nervous system arises from lateral gray column of T1 to L2 segments, thoracolumbar outflow. Parasympathetic arises from visceral efferent nuclei of 3rd, 7th, 9th, 10th cranial nerves, S2 to S4 sacral segments, craniosacral outflow. All parts of the body, somatic and visceral, receive sympathetic, and sympathetic stimulation helps body energy to fight or flight. Parasympathetic supplies only viscera, helps the body to conserve energy. Preganglionic fibers are short in sympathetic and longer in parasympathetic. Neurotransmitter is adrenergic in sympathetic, cholinergic in parasympathetic. Sympathetic nervous system dilates pupil, slows peristalsis, increases heart rate and respiratory rate, vasoconstriction, bronchodilation, pilomotor, pseudomotor. Parasympathetic nervous system constricts pupil, accelerates peristalsis, decreases heart rate and respiratory rate, bronchoconstriction.